Now that we've learned about innate immunity, let's take a look at adaptive immunity. Adaptive immunity develops a selective and therefore very targeted response to a foreign object, to recognize it and then to attack it. And then if that foreign object is encountered by our bodies in the future, our bodies are already primed to repel that specific threat. The key cellular components of the adaptive immune system are two kinds of immune cells called T cells and B cells. The body contains a pre-existing repertoire, a plethora of B cells and T cells when we're born, and collectively they have a vast array of receptors or surface markers that can recognize and bind to almost any antigen. However, at any given time, only a few cells of a given specificity are present. So, for example, at this instant of time, I may only have a few T cells that express that red receptor. So when a foreign object is detected, the body must select and make more of the correct B cells and the correct T cells that can recognize that foreign object and neutralize the threat. A part of this selection is mediated by the dendritic cells. Remember, those are those first responder cells, and they serve as the bridge, both conceptual and physical, between our innate immune system and our adaptive immune system. So after the dendritic cell phagocytoses that foreign object, say a bacterium, it can break that bacterium down and present some of the proteins from that bacterium at the dendritic cell surface, so at the end of these processes. And we call that antigen presentation. So what is that dendritic cell presenting the antigens to? Actually, your T cells. So a naive T cell that's capable of recognizing that antigen that's now presented will bind to those antigen fragments and become activated and proliferate. If a naive T cell displays the CD8 surface receptor, so that is a CD8 positive T cell, then it will become a cytotoxic T cell, and that activated cytotoxic T cell will directly attack the pathogen. On the other hand, if a naive T cell displays a CD4 surface receptor, so it's a CD4 positive T cell, then upon activation, it will become what we call a helper cell. And that helper cell won't directly attack the pathogen. It will instead help other immune cell types fight the infection. So let's talk about those other immune cells like the B cells. When a B cell encounters its compatible antigen, then the B cell will internalize it and present the antigen fragments on its surface. So you can see that this seems kind of like what a dendritic cell does. But in this case, in the adaptive immune system, this is really special because an activated helper T cell, remember those are the ones that are CD4 positive, can target the same antigen and then bind to those fragments on the B cell. This process is a really interesting and important checkpoint because it makes sure that our body says there's two kinds of cells that recognize this as a foreign object, the B cell and the T cell. That confirms the presence of the foreign object and sets off the response. And once activated, those B cells will rapidly proliferate and differentiate into either memory B cells or plasma cells. Now, those plasma cells, those cells that are within our plasma, will release large amounts of antibodies, and those antibodies are the humoral component of the adaptive immune system. Now, like the other complement system in the innate immune response, these antibodies are proteins, and these proteins are special because they have binding sites that are unique to a target antigen. We call that the epitope, and that allows them to bind to a specific antigen both strongly and selectively. And here we're showing you the schematic of an IgG antibody. Now, once released by those plasma cells, the antibodies will bind to the pathogen, and this could do one of three things. It could prevent the pathogen from entering other cells. It could flag that pathogen for attack by other immune cells in our system, or it could activate the complement system. Now, memory B cells and T cells provide a lasting immunological memory of those foreign objects. This means that when that antigen is encountered again, we're exposed again, we have another infection, this immunological memory will allow the adaptive immune response to occur faster and also in a stronger fashion. Now that we have a sketch of how the immune system operates, we can understand why immune cell therapies are attractive to treat cancer. Now remember that cancer cells are not foreign objects to our bodies, and for this reason they can often evade the immune response that a foreign object would typically elicit. So therefore, one approach has been to engineer immune cells to target the features that are unique to cancer cells, such as the surface markers on those cancer cells. The body can then recognize and kill those cells that had previously evaded the immune system and might have also escaped conventional therapy. Additionally, these re-engineered cells, for example, a CAR T cell, can persist in the body and provide that immunological memory. So now we have a high-level view of how the immune system works, at least as is relevant to understanding the applications and the challenges to cell therapy manufacturing. But let's note that we have really only scratched the surface here. The immune system is incredibly complex. We are still learning about that as an active area of study, both in its own right to figure out how our bodies respond to new threats, as well as in the applied sense for cell therapy manufacturing.